So in this lecture, we're going to look at approaches to column normalization. So from last time, we had our RNA-seq dataset, and we'll remember that every row in this dataset represents a different gene, and every column represents a different sample. And so each one of these numbers represents the number of times that a particular sequence read was mapped to a particular sample. Now, we might want to do inference on these genes, right? So, for example, we have these two samples here, these are both day 3 samples, but we might want to say, is this gene present more times in this mouse than in this mouse? But we can't do that by simply comparing these numbers, right? We can't just look at 625 reads for this gene in this mouse versus 65 reads in this mouse, because there might just be more sequences in this gene, in this sample, than in this sample. So if there's just 10 times more sequences in this sample here than here, right, then these genes aren't significantly different. It's just that there's more sequences present that we found in this sample versus the other sample. And this has to do with the way that sequences are assigned on platforms like Illumina, in which there's a stochastic number of sequences. It's not necessarily constrained that you have the same number of sequences in each sample. So you're going to have different numbers of sequences in each sample, but that doesn't reflect anything about the underlying biology of these samples. That's just something that happens randomly on the sequencers, that you just get more reads by chance that are assigned to one sample versus another. So we need to normalize in some way. Now, this is a surprisingly controversial and complicated question. So there are very heated and really passionate debates in the literature about this normalization step. And in fact, it can be very controversial for my field, for the microbiome data, about how you normalize. So there have been very heated discussions and papers that have been published in literature, such as this one from Marty and Holmes, that have argued that some methods that are commonly used to solve this problem of correcting for different sequencing depths and different samples are not valid. And we'll touch very briefly on this controversy in a few minutes. But I want to start by just thinking about the most straightforward, maybe potentially even naive way that you could address this problem, and that's simply to divide each sample by the number of reads in that sample. So we can just sum, and in R we can do this in a single line of R code. We can just sum for each sample. We can sum the number of sequences in that sample. And in R, we can do this in a single line. So here, as usual, I have my few lines of R code. I start by set my working directory to wherever I happen to have that file, to wherever I downloaded it from the GitHub repository. I read the table. Notice that I have a couple of parameters in my read table command in R. So obviously I have to have the file name. This is a tab separated file. Row.names equals 1 means that this first, there's no actual count data in this first column. The first element of each row is the name of the gene. And so this is just telling R that the name of the gene is going to be the first element of each row. And then there's also a header, right? There's no data in this top. There's no data in this top row either. Those are the names of the samples. And so this header equals true tells R that that there are sample names in the first row, and that should also not be considered as data. Now, this read table command will return a data frame. We'll remember that in R, data frames are the equivalent of spreadsheets. That data frames can have multiple vectors. Each vector can be its own data type. In this case, all the data types are numbers, so it doesn't matter the constraint that the constraint that the vectors or the columns can be different data types doesn't affect this spreadsheet because they happen to all be the same data type. They all happen to be numbers. But what R is going to guarantee is that there's the same number of elements in each row. Okay, so we have this rectangular spreadsheet, and the read table will return a data frame, again, with each vector having the same number of elements, and R will actually throw if they have different number of elements. Notice there can be missing data here, right? So if this column said NA versus 2, that would be okay with R. And notice that NA is different from 0. Right? So 0 means that there were no reads assigned here. NA would mean that we didn't know how many reads were assigned, or that data were simply missing. And that data, again, an NA is different from 0. So R unpacks the spreadsheet, and actually we can, if we're working in Windows, not directly in RStudio, but in Windows directly, we can inspect the data frame that we get. So here I said my T equals read table, and so R has figured out that the row dot names are the gene dot names, and it figured that out because row dot names equals 1 here, and it figured out that the column names are column names are the sample names because header equals true, and then our data is here as before. So again, we're seeking just the simplest possible normalization scheme to start our thinking about normalization. And so the simplest thing that we can do is to simply divide each one of these data points by the sum of all the reads in the sample. And this is just going to be straight relative abundance. So this first sample, it turns out, has 58,000 reads, right? And so we just get this by applying to our table the function sum. And this apply method is a little bit mysterious. 1 would be rows and 2 would be columns. So this is a little bit of funny R syntax, but 2 means columns. So we read this as apply to our data frame the function sum and apply to every column. And R simply spits back the number of sequences per sample. And then using, I could use another apply, that would probably be a better way to do this, but using my R code with its heavy Java accent, I just make a for loop here in order to change these raw read counts or raw counts to relative abundance. So I start with, I make a new data frame, my tnorm. This actually, in R syntax, this actually creates a new frame. In many languages, this would be a reference to the same frame, but in R, this actually creates a whole new frame. So it's copy by value. And then I'm just going to walk through every column. I'm going to take the column sum of that particular column, and then my new normalization value is each element divided by the column sum. So I have gone from having a different number of sequences per sample to forcing the sequences, forcing each sample to sum to 1. Right, so I've gotten rid of this problem of there being different sequencing depths by forcing them all to sum to 1. And when I look at this data here, right, so now instead of having 329 and 30, I have 329 and 30 divided by 580,411 sequences per sample. Notice that there are really large differences in the number of sequences per sample here, right? So this third sample from day 2, right, has almost 10-fold fewer sequences than this first one. And again, that probably doesn't reflect anything about the biology of these two mice. It's just something that happens on the sequencer. And it's not uncommon to have factor of 10, sometimes even factor of 50 or factor of 100 differences in the sample with the fewest sequences versus the sample with the greatest number of sequences. So you might ask next, well, this seems like this is a solved problem. Why is this controversial, right? You just switch to relative abundance and everything is good. But in fact, many people have problems with relative abundance normalization. And so I'll say in my lab, we actually tend to use relative abundance, these simple proportions, more than any other form of normalization. But we're actually a fairly small minority among metagenomics labs. So different people, there are many different criticisms of simple relative abundance. Although to be fair, there are criticisms of any method that you use. So there's no method that you can really safely use without getting into potential trouble with reviewers. But one common criticism of relative abundance can be found in the DSeq paper itself, which argues that relative abundance, simple relative abundance, is a highly parametric statistic that isn't robust to outliers. So the authors of the DSeq paper in their development of their normalization scheme that they use for DSeq in DSeq 1 argue that if there's a few highly and differentially expressed genes, these have strong influence on the total read count, and that will sort of distort their relative abundance. So if you have just a few highly expressed genes, they will cause all the other genes to sort of they will warp the entire relative abundance scheme and cause all of the genes to reflect the large relative abundance of the highly expressed genes. This is an example of the compositionality problem. Again, this compositionality problem haunts normalization. We are familiar with the compositionality problem from earlier in the semester, right? So we remember when we were looking at the multinomial distribution, right? And we could represent like in a three-sided die, you could represent everything you needed to know in your Dirichlet beliefs, in your Dirichlet conjugate priors, looking at a multinomial distribution. In a three-sided die, you only had 2 independent degrees of freedom in your Dirichlet, right? So if I told you my belief that you could roll a 1 and my belief that you could roll a 2, you could solve for the belief that you could roll a 3, right? So if the belief that you could roll a 1 was very, very high, right? If I'm 99% sure that I could roll a 1, then I'm pretty much close to 0% sure that I would roll a 2 or a 3. That's the same problem that the authors of DeSeq are worried about here, which is that if you have an RNA-seq experiment and 98% of your reads are related to actin, then you have no reads left over for anything else. That would skew a relative abundance or a simple normalization scheme. So there are many other criticisms of relative abundance or naive proportions, but this is certainly one that you will see consistently in the literature. So how does DeSeq in the DeSeq paper, how do they decide to what's their approach to this that they think will be more robust? So in the DeSeq paper, they are going to define these values mu. Right, so this mu here is going to be equal to some unknown value, which is the true fraction of the gene expression. Right? So Q is some value between 0 and 1, and that represents the true fraction of the gene expression, times some scaling factor, and the scaling factor is what has the number of sequences per sample. Right, so the average is mu is what you would observe. You know that. That's what you observe, but Q is the true thing. It's the thing you want to know, but it's unknown, and you have to correct for the scaling factor of each particular sample, and the scaling factor is what's going to correct for the number of sequences per sample. And so how do you estimate this scaling factor, and how do you calculate SJ? Instead of using naive proportions, the authors of DeSeq say, okay, well, why don't we use the median, right? So Kij will be the median of from a particular sample, and then why don't we normalize that by this thing called the geometric mean? And the geometric mean, you like if you have a bunch of numbers, you multiply them together, right? So here's the geometric mean of 3 numbers. You multiply them together, and then you take the cube root of those numbers. So if you have 50 of numbers, you multiply those 50 numbers together, and then you take the 50th root of that. And so this is generally considered to be a more robust alternative to average. Median is a robust alternative to mean. So clearly this seems to be and I should say it's that you're taking the median. The SJ is the median value across all of your cells. Kij is each particular read. I won't get into a lot of details about why this works, about the justification for this. I do acknowledge that it is more robust, but I'm going to make an argument here that actually, at least for our data set, and maybe our data set is one that doesn't have an outlier, so it's maybe not as sensitive to this outlier problem. It actually doesn't matter whether you use whether you use the DeSeq normalization or whether you use naive proportions. So here, just in a couple of lines of R, I'm going to implement this algorithm that's in the literature. I won't go into details about how this works, but we can see, right, this SJ is we take the median of all the reads, and we then define the geometric mean, and we just grind through and calculate the SJs here, right? But if we actually just plot the SJ is calculated by the DeSeq paper, and then this is the simple sum of all the sequences, right? So this is a scaling factor based on the scaling factor we'll use for naive proportions, and this is the SJS calculated from our DeSeq pipeline. What we see is that at least for our data set, and again, maybe our data set doesn't have outliers, so this is what makes this possible. It really, you get very, very close to the same answer from these two methods. I think there's a large theoretical difference between these methods. There's, I think the DeSeq authors are probably right, that their method is more robust, but at least for our data set, it doesn't really make much of a difference which one you use, and therefore it doesn't really seem worth getting into a large theoretical argument about which one is better. And I will say this is often true in RNA-seq data sets. So there's really a lot of support in the literature for this idea that normalization schemes don't matter very much. Perhaps my favorite example of this is this paper that was published in 2013 on RNA-seq studies. And this is looking at 5 different algorithms for differential analysis of RNA-seq. We'll be talking quite a bit about different analysis strategies. In fact, coming up on the t-test will be actually an algorithm I argue that is surprisingly robust for comparing 2 samples. Here's DE-seq. Which is the algorithm we've been talking about. And here are 4 other algorithms that these authors looked at in comparing differential expression between 2 conditions. Right? So all of these algorithms are looking at, in this case, a data set looking for significant genes between 2 conditions. There are 2 kinds of normalizations. Each one of these packages had its own normalization schemes. And here the authors are comparing it to a common normalization scheme that they've imposed on all of the packages. And what you can see here is that the number of differentially expressed genes that you get really does not depend on the normalization scheme at all. Right? So this is, I think, consistent with the argument that I just made, which is that naive proportions and DSeq, the normalization scheme there doesn't seem to matter very much. And of course, that isn't to say that there aren't going to be particular data sets where it might matter a lot, or that there aren't important differences between these algorithms. But as a matter of practice, there are many choices that you can make as a bioinformatics analyst that will impact the number of significant genes. And normalization schemes, to a first approximation, does not seem to be one of those such methods. So to finish up, let's return to our data set, our RNA-seq transcriptomics data set, and calculate these weighted averages, appropriately normalized for sequencing depth, for our conditions of 2 days, 12 weeks, and 18 weeks. So we just start in R, and you can actually copy and paste this directly into R from the PowerPoint. And so we just start defining some variables. Right, the first 3 columns of our spreadsheet are two days. The next 3 columns are 12 weeks, and the last columns are all 18 weeks. Right, so we're just telling R where we want our categories. Right, 3 columns of 2 days, 3 columns. Go back to the original spreadsheet. 3 columns of 2 days, 3 columns of 12 weeks, and the rest at 18 weeks. Again, confusingly starting with W20 instead of W18. But that's fine. We'll get the columns correct in R. We set our working directory to wherever this file is. Read this in as a table. Again, separating the columns by separating all the fields by tab delimitation. This is a tab delimited file, and so we just tell R that this is a tab delimited file. We have our row and header information. So the data are the gene names and the sample names. Here I'm just going to add, because I'm taking a geometric mean, I can't have any zeros in the spreadsheet, right? If you can't have a 0 as you take the product across all your data. So I just add a pseudo count of 1, and then I calculate my medians, calculate the SJs according to the formula in R, and then this is just grinding through. So if you know SJ, right, this Kij is the count of each cell in the spreadsheet, and this is the what we want is the average normalized value across conditions. So for 2 days we have 3 conditions. We're going to sum across the 3 conditions, normalized by the scaling factor in each sample, and then normalize divide by 3 because we're taking the sum of 3 and we want the average expression in these 3 conditions. So we just grind through this in our R code. And then we calculate these weighted averages for 2 days, 12 weeks, and 18 weeks. Right? And so when we do this, we can make a data frame that holds all of this information. Right? So I have my 2 days, 12 weeks, and 18 weeks. I make a new data frame and call that params. And then again, this is what this is going to look like. So now for my first gene in the data set, these are the weighted averages for 2 days, 12 weeks, and 18 weeks. And these have now been normalized by sequencing depth because in the DeSeq universe, this Sj is the scaling factor. Okay, so this is one approach to normalization. If that seems overly complicated, and you know it's maybe it's overly complicated, maybe it's good to have that it being complicated because it's robust to outliers, but it's very, very similar to just taking relative abundance and then scaling to some number other than one in the normalization. So that's fine. So we have our normalized data correcting for sequencing depth at 2 days, 12 weeks, and 18 weeks. If we just look at without normalization, right? So here I'm just plotting the raw data on a log scale, right? So on the x-axis I have the 2-day data, right? So I'm taking the mean of each of my 3 samples at 2 days, right? The mean of each of my samples at 2 days, and log 10 with a pseudo count. So this is the mean of each of my samples at 2 days. This is the mean of each of my samples at 12 weeks, right? And I can see that without normalization, I have really serious problems, right? So this red line is the identity line, right? And so I'm not ready to do inference on 12 weeks versus 2 days, because that inference would find every single one of these genes significantly different from the red line, right? But that isn't because there's anything biological necessarily at 12 weeks versus 2 days. This is just that there happen to be more sequences at 12 weeks versus 2 days with a relatively small sample size, and so we need to correct for that in some way. So this is with the out normalization, and this is with the DeSeq normalization, which again is going to be very similar on this data set to simple naive proportions, and we're now much more centered along the red line with right. So we're much more able to do successful inference here comparing to the red line, which is that the expression of the gene is the same for 2 days and 12 weeks. Now notice there is this sort of region here where maybe we sort of drift off the red line, and so this gets to be a little bit of a questionable region where do we really want to say for the highly expressed genes that all the highly expressed genes are going to be higher at 2 days than at 12 weeks? So maybe instead of using a simple normalization like DeSeq used, maybe we want to use some kind of local regression that would draw the red line of our null hypothesis along this region here. And again, you can see where this gets to be very controversial, and where there's going to be many, many different ideas about how to do these kinds of normalizations in the literature. And again, this is a very rich area of the literature that you could spend a lot of time looking at. Now, okay, so that's sort of the normalized means. You could say, well, what about the variance, right? And you might think that the variance would, within a condition, would just be something like, so if I wanted the variance of all the genes at 12 weeks, right, that this worked for mean, that I could just apply, I could just take my spreadsheet, look at the columns that were defined as my 12-week columns. So remember that 12 weeks is just the columns that are defined at 12 weeks, and just use canonical variance the way that I did canonical mean. But there's all kinds of reasons why actually this is not a good enough variance for DeSeq. And the first of these is actually pretty easy to understand, which is remember that the negative binomial distribution, which is what DeSeq is using for its inference, requires that it not be true that the variance be less than the mean. Right? So we had our 2 parameters for the negative binomial distribution that you guys will remember, right? And we could express these 2 parameters in terms of the mean and the variance, and you can rearrange these to get our equation of the relationship between the mean and the variance in the negative binomial distribution. So this is the observed variance under the negative binomial distribution. This is the mean of the samples, right? And remember, we were thinking about in the negative binomial distribution that R is related to the tournament length. So when R is infinitely long, when you have an infinitely long tournament, it's just like flipping a coin over and over again, right? In that case, you have essentially a Poisson distribution if the probability of success is small, and under the Poisson distribution, the mean equals the variance. But when R is shorter, when the tournament is not infinitely long, then you have you've added additional variance, and in this way you can allow for overdispersion. You can allow for the variance to be greater than the mean. But these equations have no way for the variance to be less than the mean. So if you calculate simply the variance of each gene, it's going to be true in some cases that the variance is less than the mean, and you won't be able to fit the negative binomial distribution. Okay, so you have to define the variance as the mean plus something. You can't just use simple canonical variance. And so this causes DeSeq to define variance in this way, where we have what DeSeq will call the shot noise, which is the variance would be under the Poisson distribution, and then what DeSeq is going to call the raw variance is the extra variance that's associated with the negative binomial distribution. Right? So the raw variance here equals the canonical variance weighted somehow for normalization, weighted for sequencing depth. But this is related to canonical variance. This is the variance that we would expect to see. And DSeq is going to define this raw variance as this canonical variance minus the weighted mean, which is again the variance that you expect out of the Poisson distribution. So we'll look at this in the next lecture. We'll actually take a look at taking our simple data set or our RNA transcription data set through DSeq 1 and thinking about the way that the variance relates to inference.